All right, let's get it in. Goes to Kennewick Man recap. The history preceding this 2018 study. Really, this was a preprint that I was reading from, but nonetheless, the complete, I guess you could say, the final publication came out in 2018. What made it so controversial? In the previous study of Kennewick Man in 2015, geneticists concluded that Kennewick Man's DNA was closely related to modern Native Americans and thus supporting the One Foundation population model. All right, so in part one, I went over the background of the two molecular geneticists that were involved in this study, and I went over their thoughts pertaining to mainstream methods in terms of sequencing DNA versus what they recommended and what they used in this particular study. And so I'm going to recap that. Okay. Dejian, Yuan, and Huang, I think I'm pronouncing that correctly, were the authors of the study titled on the peopling of the Americas, molecular evidence for the Paleoamerican and the Solutrean models. Criticized the standard method of sequencing DNA, which is the genome-wide method that focuses on fast SNPs. So the genome-wide method is what mainstream geneticists typically use. They criticized it basically because the genome-wide method is basically an averaging of fast SNPs across the genome, basically. And I'm going to get into more detail as the slides progress. What are fast SNPs? Fast SNPs are SNP sites that mutate rapidly. What was the specific criticism of the fast SNP methods, or method, I should say? Due to the fact that fast SNPs mutate rapidly, they are commonly susceptible to mutation saturation. Susceptible to sudden environmental change, which was characteristic of the ancient world. The reason why I said that is because as it is historically documented, they say that 25,000 years ago was the last glacial maximum. And so there was significant climate change before the last glacial maximum and after as well, And so as the weather progressed, the environment changed, that could theoretically affect fast SNPs because they are dictated by environmental changes. As a result, many population groups can share many fast adapting SNPs, giving the impression that these groups share deep ancestry while in reality only sharing recently evolved SNPs due to rapid and or harsh environmental changes as opposed to naturally selective SNPs that are a true reflection of natural selection and or drift, which is symptomatic of true biological divergence. These SNPs evolve independently of biological evolution. Therefore, two unrelated populations can appear to be much more closely related than they actually are. This is why Dejian, Yuan, and Huang labeled the fast SNP method of mathematical illusion. Examples of fast evolving SNPs. That would be depigmentation alleles, lactose tolerance, high altitude adaptation, immunity, diet, and metabolism, inherent to morphology. What is mutation saturation? Mutation saturation occurs when a DNA position mutates so many times that the present day state no longer accurately records the full history of changes that occurred at that site. Odometer analogy. You can think of a mutation saturation the same way that you would imagine an odometer reaching its mileage capacity, meaning every odometer has a digitalized limit in terms of placeholders. Your odometer may only have the capacity for 6 digits. So the capacity limit would be, this is theoretical, I don't even know if odometers go up to 999,999 miles. But just as an example, anything exceeding 999,999 miles would not be recorded, and therefore the true history of the mileage of that particular vehicle could be masked. The vehicle could be recording 999,999 miles, but may actually be well over that. In terms of genetic mutation, once saturation occurs, mutation timing can be convoluted, which can give the appearance that two population groups are more or less related than they truly are. To combat this, Dejian, Yuan, and Huang proposed the slow S&P method. This method yielded completely different results. What's the difference and how do these methods impact results? What are slow S&Ps? These are S&P sites that mutate slowly, not as susceptible to rapid change from environmental factors. Since these S&Ps are evolved slowly, they preserve ancestral DNA at a much higher success rate and are a more accurate reflection of descendant divergence through drift and or natural selection. Advantages. Slow S&P sites preserve mutations much longer and potentially retain deep ancestral signals. What were the criticisms of using the slow S&P methods, and how were these criticisms addressed by Dejian, Yuan, and Huang? This is out of context. Let's see some. I think that should be right there. After advantages, we'll start right there. Okay, so this is just an illustration right here. I had AI put this together, of course, but it's just a brief illustration to demonstrate how fast S&P methods can aid in smoothing narratives based on these two geneticists' model. So you can see here that basically what fast S&Ps do to genetic data is basically compress genetic data, and it can convolute what populations are related to what, how far in distance they are from each other, and so forth and so on. And I think the illustration pretty much speaks for itself. Thought that was nicely done by ChatGPT. And you can see number 5, the box number 5, you can see it says lost ancestors. When data are smooth, deep ancestry becomes invisible. Okay. Number 4 says genetic iron. Before smoothing, high resolution detail and rugged. Smoothing reduces genetic resolution. After smoothing, low resolution, flatten, and simplify. What were the criticisms of using the slow S&P methods, and how were these criticisms addressed by Dejian, Yuan, and Huang? All right. Criticizing for filtering of fast S&Ps. mainstream argument. That's bottom right there. I got to straighten that up, man. Yeah. Okay, they criticize him for the filtering of fast S&Ps. Mainstream argument. Genomic information loss. Filtering fast S&Ps only captures deep ancestry that may be shared in deep time, but not necessarily in ancient America. All right, so counter argument, and basically how these two geneticists basically address this is mainstream detractors never replicated Dejian. Well, let me do it one at a time. Genomic information loss. They accused these two geneticists of, theoretically losing genomic information. My own counter argument to this is that mainstream detractors never replicated Dejian, Yuan, and Huang's method to directly measure loss. So basically, that's a hypothetical claim that hasn't been proven, is what I'm trying to demonstrate here. But this next one, the next point, is proven within the study in and of itself. The next point is filtering fast S&Ps only capture deep ancestry that may be shared in deep time, but not necessarily in ancient America. The counter argument based on the genetic study is signals were traced from the most ancient populations, which was, if I'm pronouncing this right, Mesmachia. This was found, that sample or specimen was found in Russia, in ancient Russia. To intermediate populations such as Anzac 1 and Kennewick Man, I believe I got those ages right. To modern populations in North America, like the Mexicans and the Mexican Los Angeles population, that I'm going to go into detail about. All right. Let's see here. My computer beeping. Maybe somebody up in here. Let's see. It's something else. Okay, back to it. Okay, African signal, mainstream argument, just a shared ancestral artifact. In the publication, they pointed out that the African portion of the archaic African component found in Kennewick Man was shared the most affinity to the Mabuti. I think I'm pronouncing it correctly. Mainstream argument is that it was just a shared ancestral artifact. Counter argument is the signal was consistent from ancient populations to modern Native American populations. Not all Native American populations shared the signal. The signal was absent in some Native American populations. If the signal was simply an ancestral artifact, then the signal should be evenly distributed amongst all Native American populations. Normally how it goes. Population panel summary. I think these visuals are going to be cool. I had AI put this together in terms of the panels that were used and the populations within these panels that were used. Okay, this is from the 1,000 genome reference population. And I ain't going to read them all. It is simply displayed on the screen. And I'm going to keep going. Human Genome Diversity Project. These are the populations that were included in the Human Genome Diversity Project panel that they in particularly used. So a total of 13 there. Let me see. I didn't have a total. A total of 13 there. South Pacific and Australasian reference populations used by Huang, Huang, or Yuan and Huang. Pretty much self-explanatory here. So a total of 7 from that reference point. All right, archaic human specimens used by Huang, or Yuan and Huang. Okay, and so once again, this is abstracted from the actual study by AI. I think this is pretty thorough. Little fly visuals. Ancient Eurasian specimens used by Huang. Okay, ancient American specimens. I wasn't too far off of my age estimate for Anzac 1 and Kennewick Man. Three of those specimens were used. And this is the overall populations and panels that were used in this study. So you had 38 modern populations, 14 ancient human specimens, and 3 archaic human specimens, for a total of 55. I think the prior study in 2015 had about 52. Indications of results, relative affinity charts. Again, this is AI derived. These numbers, I'm not saying that these are, something that was included verbatim in the report, but these numbers are derived from the report by AI, So I'm not trying to say that these are the words of these two geneticists exactly, but it demonstrates what they were conveying in terms of affinity relationships. So here we go. Okay, so this first chart represents Kennewick Man's overall affinity to all populations involved in the study. And you can see number 1, you have the Peruvians from Lima in South America as sharing the strongest affinity. You have the Kiritiana second, the Surai, I think I'm pronouncing that right, third, Mesmakaya fourth, the Mamanawa, which are Philippine Negritos, fifth, Papuan sixth, seventh is Aboriginal Australians, number 8 is Yorubans. All right. So one of the most important things that I want to point out here is particularly the Kiritiana and the Surai. They keep popping up in these ancient studies. And if anybody can remember the Australian signal study, the Y population signal study, they popped up in that study as well. Having an Australian signal that wasn't present upstream in Central America, wasn't present in, was it Mexico? Well, I said Central America, North America, including Canada, up into the Bering Strait region, Siberia, Central Asia, or Central Eurasia, or West Eurasia. So pretty much the only place that signal was found in the world that they know of, or at least they found to date, is in these two Amazon populations. So here again, Kennewick Man is sharing a high affinity with these two populations again. Just keep that in mind. Weakest affinities. Russians. This is interesting because obviously these are modern Russians, because the sample or the specimen that Kennewick Man showed the most affinity to is from Russia. So, as I move forward, we're going to find out what other characteristics of Kennewick Man in terms of his autosomes that he showed. And I'm going to put this together. So just keep in mind that he didn't share affinity with modern Russians. This is going to come into play later. And pretty much all East Asians, he didn't share affinity with. All right. What's also important is that he didn't show affinity with Denisovans. And this is what the authors of this study used to support the Solutrean model. Like I said, I think I'm pronouncing that correctly, because if they're saying if the migration was from Eurasia, whether it was West or Central Eurasia to the east through the Bering Strait, they're implying that they should have had more genetic contact with Denisovans. But the archaic signal was shared more affinity with Neanderthals. So that's another point that they build this conclusion of the Solutrean model with, based on those genetic findings. All right, so this is the African associated signal that I was hitting at earlier, the summary of that. Okay, so the archaic specimen that shared the most African archaic signal was the Mesmakaya 1, which was found in Russia. Secondly, the Yorubans, then the Mota, and again the Peruvian Caribbeanos and the Suriai, modern day Native Americans, right? Then you got the Mamanwa from the Philippine Negritos, or the Philippine Negritos and the Papuans. Again, weakest affinities found in this study. Han Chinese, Japanese, Russian again. So this is how we know that this is a different Russian. These are melanated Russians, because this is a Russian. This was a specimen found in ancient Russian. The date was estimated to be 65,000 years ago. Okay, so this idea that melanated people weren't in all parts of the world and land masses at the bare minimum before the existence of these depigmentation alleles, and basically the existence of I'm not gonna say non-melanated populations, but lesser melanated populations. I'll put it like that to be the most accurate, right? So, and not just melanation, but the differences morphologically, between populations. You can see who was in the region in ancient times. Once again, melanated. They call this signal within this Siberian, archaic Siberian. They're calling it an archaic African signal. So just know that, haters. Some things you just have to accept. You know what I mean? We gotta. You guys gotta. The haters gotta stop doing that to themselves, man. Gotta stop denying facts, man. And even if you don't accept everything within this study, these things, in terms of melanated individuals being the most ancient populations, the first, the indigenous to all land masses, that's consistent amongst all genetic studies. Okay, so moving forward. Okay, what's also important is the West Eurasian affinity summary, with Mesmeriah being first in terms of affinity with Kennewick Man. You got the Aussie, Stuttgart, the Lebrana, Lashbor, Kostenki, 14, the Vestanese, and the Elmoran. All right, the Elmoran Affinity Index. Relative affinity analysis. Elmoran was the most affinity, shared the most affinities with Anzac 1. Positive correlation with the modern Mexican Los Angeles population. General Native American cluster. Shared positive subsets with the Peruvians. The Colombian, I think that's Colombians, Puerto Ricans. Kennewick Man is number 7. Then it had a negative correlation with the Mexican Los Angeles population. And I want to explain that later on. It's very important because with that negative affinity, they measured that negative, or they isolated that negative affinity, and they found an additional African signal. But again, I'm gonna get into that once we get to that part. Okay, so okay, so that's just a bar chart underneath that to give you another visual effect there to hammer home the point. Okay. What's important here as well is over here, proposed relationship. So they're proposing that the Elmoran is ancestral to Anzac 1. Anzac 1 is ancestral to certain Native American populations and its modern descendants. Okay, and that's all going to come into play later on when I explain this negative affinity. I'm going to put this all together. Because basically what they're saying is modern Native Americans are most related to, or share the most affinity with, Anzac 1. All right. And Anzac 1 shares the most affinity with Elmoran. Within the Elmoran population, there was a archaic African signal, very similar to Kennewick Man, right? But they found another African signal. And like I said, I'm going to explain that when we get to that part. It's very interesting, man. This is thorough, man. Got to give it to him, man. Okay, this is Anzac. Shared the most affinity with Native Americans, just like I said. Second, Elmoran, the Caratiana, Surai, the Peruvians from Lima, Kennewick Man number 6, the Mamanwan Philippines population, Colombians, Puerto Ricans, and then the Yorubans, right? Let's move forward. African affinity of Elmoran, Anzac, and Kennewick Man. All right, so Kennewick had the most African affinity. Seems like Anzac had the second most, which is also very interesting. And I'm going to explain that as I move forward. That ties into what I've been talking about. Okay. And they offer an explanation as to why. Because if you see here, if the Elmoran is ancestral to the Anzac, right? But the Anzac, now let's put this together. Kennewick Man had the most affinity with Africans, right? Anzac is second. Elmoran is third, right? So if Kennewick Man is has the elder ancestry in that specimen that shared the most African affinity within the Kennewick Man's African genome is 65,000 years old. And El Miron shares a similar signal, but it's lower than Anzic. This prompted, and this pattern was shown in modern day populations, which prompted the two geneticists to answer the question as to why the African affinity goes up in the descendant versus the ancestor or the intermediate ancestor in El Miron, right? If y'all following me. Okay, Native American population signal comparison. This is a good visual as well because it's showing you very simply who have what affinity with what and what type of affinity. The Terratiana showed once again, which is consistent with that 2015 study, an Australo-Melanesian signal. It had an African-associated Mesmachia related signal. That's the specimen from Russia that's 65,000 years of age, or existed 65,000 years ago, based on their estimates. The two groups from the Amazon carried both signals. That is very significant. That cannot be ignored. Because again, in the 2015 study, it had, it was cited to have the Y population signal, which was Australian as well. And then it had, they also show in this study, the Kennewick Man, or at least the Mesmachia related signal. All right, which is basically Kennewick Man, at least the Archaic African portion of his genome, right? Kennewick Man shows Austro-Melanesian related signals as well as African-associated Mesmachia related signals. He carried both as well. Very interesting. All right, Mesoamerican groups they shared more affinity with Australo-Melanesians. North American indigenous Paima, more affinity with Australo-Melanesians. Also, too, man, I want to point out too that the second most prominent Y-DNA haplogroup amongst modern Native Americans is Melanesian derived. I think that Y-DNA haplogroup is either C2 or C3. Obviously, all C related haplogroups are Australo-Melanesian derived. Okay, that's in their normal ancestry. That's mainstream news. That's not alternative science or anything like that. That's mainstream news. So you got to ask yourself, if one of their main Y-DNA Y-DNA haplogroups are Melanesian, you know what I'm saying? Like, couldn't the rest of this be pretty plausible? I'm just saying. Okay, moving on. But the North American indigenous Paima didn't share any African-associated, at least ancient African-associated signals. All right. South American reference, the Peruvians shared African-related signals. All right. That was related to the El Miron. Okay, but they didn't share it as much as the Mexican population. Like I said, I'm going to go over that in more detail as I go. North American ancient Anzic 1 didn't have any Austro-Melanesian related signals or African-associated Mesmachia signals. Sackett had none of either. And I guess these question marks really mean not clearly. No clear signal. Not that it was none, it's just no clear signal. Diluted. All right, and the X means none at all. And it was clearly negative. It was with the X, the red X. Okay, so let's go. Kennewick Man's DNA yielded 3 affinities. African archaic type. Okay, again, that was found in the Mesmachia specimen. Had Australo-Melanesian DNA and Western European, all of which we just went over in the past slides. The archaic African signal was the most related to a Neanderthal specimen found in Russia, Mesmetskaya. Again, this signal predates West African population signals by 62,000 years. I'm just pointing that out to keep it in perspective of different, African related signals. Everybody wants to associate everything with a West African signal, but there is something more. There are African signals more ancestral to West African populations. Just putting that out there. Similar Y population signal. Australian signal found in the same populations observed in a 2015 study by, I think Scullin is his name. I might have that wrong. As far as the spelling it. But y'all get what I'm saying. That's the same thing I was talking about earlier. Other affinity relationships. Anzac One populations show more affinity to El Miron populations than East Asians and or Siberian populations of that time period. That's interesting. So here we, in the prior slides, it said the Anzac One populations showed affinity to Native Americans. Shows affinity right here is saying it's showing affinity to El Miron populations, but more than East Asians or Siberian populations at that time period, which was between 15,000 and 20,000 years ago, which is supposed to be the the beginning of the differentiation of the Native American ancestors going through Beringia. El Miron had a similar archaic African signal. All right, populations with negative affinity to El Miron. This is what I was talking about earlier, and this is gonna, make this make sense and, explain how this is relevant and how this is significant at the same time. This was another African-related signal discovered. Right? Mexican populations in Los Angeles were determined that they partially derived from an African-like population indigenous to the Americas. How was this determined? Through El Miron and Anzac One connection. This was determined through multiple factors on the same linear ancestral line. They demonstrated strong affinity between El Miron and Anzac One populations, thus determining that El Miron as an ancestor to Anzac One. It was previously determined that El Miron had archaic African-like properties. When compared to modern populations, most Mexican Los Angeles populations share positive affinity with El Miron, at least in the African components, right? However, some populations showed significant negative affinity to El Miron, indicating later admixture that the El Miron population encountered upon entering America. Once that negative affinity was observed in the Mexican Los Angeles population, negative affinity was isolated, and an additional African-like signal was observed that shared the most affinity to modern West Africa. The study said, quote, "We further found that the MXL samples with negative correlation to El Miron were closer to present-day Africans in Kennewick Man, but more distant to El Miron and Lebrano 1, with El Miron far more distant than Lebrano 1. This pattern suggests El Miron related people upon landing in North America had admixed with African-like Amerindians who had settled earlier, therefore resulting in offspring with less El Miron and more African genomes." This is a direct quote. I got the page on there. It's on page 6. So I'm going to repeat that. We further found that the Mexican Los Angeles samples with negative correlation to El Miron were closer to present-day Africans in Kennewick Man, but more distant to El Miron and Lebrano 1, with El Miron far more distant than Lebrano 1. This pattern suggests that El Miron-related people upon landing in North America had admixed with African-like Amerindians who had settled earlier. therefore resulting in offspring with less El-Miron and more African genomes. Again, this is on page 6. So we already know that El-Miron is ancient. All right. It's saying that this signal entered, the signal was already present before this El-Miron population entered the Americans based on their model and theory. All right. Whether you agree with it or not, I'm just explaining it. You know what I mean? And so they rule out any chance of these two African signals being of recent transatlantic slave trade. These results confirm that the African ancestry in the Peruvian Lyman individuals is different from those in the Puerto Rican and Colombian individuals. All right, this part is significant because it's saying that this second signal is not related to the third signal that they are associating with the transatlantic slave trade. They're saying that the two signals are different. Okay. This puts a strain on the transatlantic slave trade narrative. Why? Because the modern West African signal found in the Mexican Los Angeles population was indicated to be present in Americas prior to European arrival or transatlantic slave trade. And again, if you remember, they're calling that signal West African-like. Again, you know, these titles for these genomes are simply that. They are titles based on modern populations from these modern panels. Okay, and the narrative is that West Africans are ancestral to Black Americans, right? But here we have the so-called West African signal present in America, at least based on this model, present in America before European arrival, at least contemporary European arrival and transatlantic slave trade. In addition to that, Puerto Rican, I know I slaughtered the spelling of Puerto Rican, but the Puerto Rican signal was identified as a signal associated with the transatlantic slave trade, but yet different from the modern West African signal present in the Mexican Los Angeles reference. So, you know, my Mexican peoples, man, you know, at least some of y'all, I can't say all of y'all, you know, you're gonna try to bang on a whole population group because of what, you know, some people said online, but you know, at least within the spaces that I'm a part of, there's often a lot of criticism when I state the fact that Mexican populations, whether it's North America or whether it's Central America, including Mexico, they share, you know, African ancestry. This has popped up in this study, it's popped up in several others. But when we say this, you know, oh no, you know, y'all just wannabes and blah, blah, blah. Now, some of them will accept it and they'll say, you know, it came by way of the slave trade. But here it is. This is indicating that one of the signals is associated with slave trade. The other modern signal was not. So if all modern signals associated with West Africans are supposed to be from the slave trade, then why in North America it's not showing it? But in, you know, that was South America. That would be South America that it showed the signal in. Oh, I'm sorry. No, this is the Puerto Ricans are the ones who showed that signal, right? So how could this be, right? It's supposed to be the same signal. Logically, these two signals should be almost identical based on the slave trade narrative because theoretically, all Black Americans were supposed to be abstracts to modern West Africans, but this study cited two different modern African signals. Unexpected haplotype relationships. This is important as well. Kennewick's mtDNA. Kennewick Man carried X2A. X2A was carried into the Americas by the Solutrean migrators. At least that's what they concluded. All right. Within X2A, the SMP 1719 is found common in Denisovans and Australo-Melanesians, and the X2A specific SMP 14502 is found common in Australo-Melanesian, South Asians, and Han Chinese. The authors concluded that this was due to Solutrean populations admixing with population populations within ancient America carrying Austro-Melanesian and South Pacific ancestry. This conclusion is also consistent with X2's distribution pattern. Okay, the reason why I added that part in because the distribution part for X2A pattern is East Coast prevalent. You know what I mean? It's clustered on the East Coast, and that's supposed to be the coastline where this Solutrean population, migrated into. You know, again, the X2A is not found anywhere outside of North America. Meaning it's not found in Canada, it's not found in Alaska, it's not found in Beringia, it's not found in Siberia, it's not found in anywhere in the world besides North America. So if it got here, the question that obviously needs to be answered is, how did we get here? They offered their explanation here. And it makes sense because of the way that the X2A is clustered and patterned on the East Coast. All right. So people don't have to accept these findings, man. But what you should do, if you have any type of integrity about yourself, you should be trying to offer plausible explanations as an alternative to these findings. Instead of just name calling and attacking people personally, you should have some explanations to explain this. But people who, attack people, make, personal attacks and jabs and, create memes of you and everything like that on Facebook and everything like that, they often have no credible explanations for these anomalies. But, on the side of this Black indigenous community, we often do. So moving forward, 4 of the SNPs found in A2 are also found in non-A-type haplogroups. And the only SNP associated with A2 that is specific to A2, which is A27, or 8027, I should say, is also found frequently in L1C. mtDNA C1, C4C, and D1 share more affinity with Neanderthals than other C and D types, which the authors concluded was due to Kennewick's archaic African ancestry. Kennewick's Y-DNA. Kennewick Man shared high affinity with Malta 1 within its European allosomes, which would also suggests that Kennewick Man may have had Siberian ancestry as well. Malta 1 was found to be more related to contemporary Europeans than to Amerindians, which is a contrast from the previous 2015 study. Second African signal explained by mtDNA LC1. I think I already explained that. Yeah, I already went over that. I think that's a slide that I meant to take out. But anyway, the Austro-Melanesian signal was not recent admixture. So where did it come from? This is where I will pivot into another, I will segue this into another segment that'll be an offshoot of the Kennewick Man series, because this is related to West African ancestry. It ties in as well to melanated Europe and Oceania in a way. All three. I'm gonna tie them all together, and it's gonna be non-subjective because it's a very non-subjective. There's 2 very non-subjective reasons as to why that population was deeply melanated. Now, you already demonstrated the specimen that was found in ancient Russia and its African archaic, DNA, right? So take it over to Europe, further west into Europe, and I'm gonna demonstrate how that all ties in. Again, West African ancestry, ancient, Europe, Oceania. It's all tied together, man. Because, like I said before in my previous presentations, man, the gene flow is not coming out of Africa. It's present in Africa now, but that gene flow came from other places, man. And that's what these specimens are showing. But I ain't gonna let it all out the bag, man. I'm gonna show you when I show y'all. Yeah, moving forward. This study demonstrates that the mainstream simple conclusion of simple Native American genetic continuity by unilateral markers alone was not enough to determine the full ancestry of the Kennewick Man's population group. African Australo-Melanesian morphology correlated with DNA results. 1-to-1 was more related to contemporary Europeans than to contemporary Amerindians. The first DNA sequence study conducted in 2015 used a narrower population panel than did the 2018 study, at least in terms of some of the ancient populations. So, it wasn't like a wide margin, but they did use significant, a handful of extra, ancient specimens that the 2015 study didn't use. Which is more than likely a major contributor in the difference in the conclusions of the autosomal makeup of Kennewick Man, with the difference in S&P methods seemingly contributing to the largest difference in results. This is a quote from the actual study, from the conclusion, in their own words. Our results suggest that Southwest Europeans of 15,000 to 20,000 years ago, such as the El Miron, had a special genetic connection with the Clovis people and Yuan in North America, more so than East Europeans and Siberians of that age. Consistent with leaving Europe in ancient times, the genomes of these people are largely absent in present-day Europeans. That's very important. And the reason why it's important is because we know present-day Europeans, they're predominantly, white Caucasian, so to speak, you know what I mean? They're saying that, that these ancient Europeans don't share affinity with modern Europeans. Very consistent with what I've been saying, all right? Which is unlike all other ancient European genomes examined here. Such results suggest that okay, so let me back that up. They're being specific to Anzic. They're saying that the rest of the European genome does show some affinity with modern Europeans, but I'm explaining what way, because it's not direct affinity. Okay, I've been saying that's consistent with what I said. You know what I mean? And I'm gonna explain all of that in these segues. But such results suggest that the part of ancestry in Anzic-1 that is shared with El Miron can only have come from Europeans who had come to North America by way of the Atlantic Ocean. Therefore, people of the Clovis culture appear to be the progeny of Europeans and the first South Americans. This explains, now that's totally different from the out of Siberia migration theory, because they're saying that they're strictly out of Siberia, passing the Thuringia, going into Alaska, and you know the whole spiel. But here they're saying that the Clovis is descendants of these ancient Europeans and the first South Americans. This explains the morphological finding that the first North Americans show trace in between modern Australo-Melanesians and Europeans 1 through 3. So I think it's talking about the figure, illustrations when it says 1 through 3 from the report. Our results also showed a fraction of present-day Native Americans to share more ancestry with Kennewick Man or El Miron. Some North Native Americans show replacement of El Miron genomes with Kennewick Man related African genomes, consistent with the first North Americans occupying an unresolved morphological position between minor South Pacific and European populations. These results could not be found when using adaptive, fast-changing DNA sequences, because present-day people living in the same location may share adaptive sequences despite different ancestry. It must be emphasized that any result must be held as uncertain at best until it has been verified by at least one other independent approach, unless the result is a logical deduction from self evident exomes. Previous molecular results based on unrealistic assumptions have routinely contradicted trait evidence and common sense. In striking contrast, the results from our new molecular method, both here and in previous work, 19, have consistently found unity between molecules and traits. I'm going to go down here and make sure this is maybe change the font on this so we can fit all this in here. Take it down to 18. There we go. Let me read that last part. Okay. So it says these results could not be found when using adaptive fast changing DNA sequences because present-day people living in the same location may share adaptive sequences despite different ancestry. It must be emphasized that any result must be held as uncertain at best until it has been verified by at least one other independent approach, unless the result is a logical deduction from self-evident axioms. Previous molecular results based on unrealistic assumptions have routinely contradicted trait evidence and common sense. In striking contrast, the results from our new molecular method, both here and in previous work, have consistently found unity between molecules and traits. We expect this intellectually satisfying pattern to continue to hold in future studies of other evolutionary puzzles. All right, man. And that's the end of that presentation, man. And I just want to say something to the people, man. Let me get myself back up here. Let me take this screen on down. Bang. That's it, man. I don't see any viewers. I don't care. I think I did my thing, man. I'm gonna pat myself on the back for that. I feel like I'm doing my individual duty, man. Everybody got to do their individual duty in this movement, man. We got to make sure that the momentum keeps rolling, man. So with that being said, man, I'm gonna give myself a round of applause. Where is that, man? Yeah, back it on up, man. One more time. So, with that being said, man, the basic conclusion to what these two geneticists are saying, to put extra emphasis on it, is that all human populations, generally speaking, are going to share fast adaptive SNPs within their local region, and sometimes it doesn't even have to be local. You know, if environmental factors are the same, whether it's in the same local region or not, populations can share these same SNPs, and based on the genome method that mainstream, geneticists use, they're going to pick up predominantly on these fast adaptive SNPs, which is going to convolute the genetic results. So this right here, I hope to be, an assistant in basically cracking this genetic code and helping, these geneticists lay this foundation for cracking that code and basically bringing light to how, this DNA is being manipulated. I can't say that every geneticist is purposely manipulating the data, but one thing I can definitely say is that the foundation of the institutions that are basically educating these geneticists have got to be manipulating the information. You know what I mean? Because if you think about it, it just doesn't make sense. How can you differentiate ancestry within populations by using SNPs that, population share at a rate that is going to convolute individual ancestries of these individual populations, man. You know, I think I've explained my point thoroughly. And with that being said, we'll close it out. And like I said, this is going to lay the foundation indirectly, leading into ancient Europe, West African ancestry, and Oceania. I'm going to explain how all this is, related together. You know what I mean? Because I received criticism in the past for saying this, but I cited a reference study. Well, I referenced a genetic study when I said that. You know what I mean? But to detractors, it don't matter to them. But who cares? I'm gonna keep pumping it out like speed. So peace.